The overall goal of this procedure is to obtain image series from a mobilized human sperm labeled with a fluorescent calcium reporter dye. First, motile cells are isolated from seminal plasma by swimming them up into incubation medium. After capacitation, cells are labeled with fluorescent calcium indicator, then transferred to a perfusible imaging chamber which is mounted on an inverted fluorescence microscope. Calcium ion concentration responses are captured as a time-lapse image series and analyzed to extract single-cell responses.
Hi, I'm Edwin Andreas Morales Garcias from the laboratory of Dr. Stephen J. Publikova in the School of Biosciences at the University of Birmingham, UK.
I'm Catherine Nash, also from the Publikova lab.
And I'm Jennifer Morris from the Publikova lab.
Today we'll show you a procedure for imaging intracellular calcium responses in individual human spermatozoa.
We use this procedure within our laboratory to study regulation of sperm activities using stimuli derived from the egg and the female tract.
So let's get started.
Store semen samples at 37 degrees Celsius for no more than 30 minutes from the moment of collection. To isolate motile cells from the seminal plasma, pipette 1 ml of Earls balanced salt solution supplemented with 0.3% BSA, abbreviated as SEBSS, into a series of 5 ml polystyrene round-bottom tubes. Gently underlayer the SEBSS in each tube with 0.3 ml of semen and cap the tube loosely. Incubate the tubes at a 45-degree angle for 1 hour at 37 degrees Celsius and 6% carbon dioxide to allow motile cells to swim up into the SEBSS. Gently pipette off the top 0.7 ml of solution and pour the solution from all tubes into a clean 15 ml polystyrene centrifuge tube. Shake gently to mix. Count cells that have been immobilized with formalin in a Newbauer counting chamber. Bring the concentration to 6 million cells per milliliter in SEBSS and divide the sperm into 200 microliter aliquots in 5 ml polystyrene round bottom tubes. Cap loosely. If the response of capacitated cells is being studied, incubate the tubes for 5 to 6 hours at 37 degrees Celsius and 6% carbon dioxide to allow capacitation. Apply 10 microliters of 10% poly-D-lysine as a number of small drops to the center of a 22 by 50 mm No. 1 cover slip and allow it to dry completely. Use vacuum grease to attach the cover slip, treated side up, to a perfusible imaging chamber built for the purpose. For each imaging experiment, select a single tube of capacitated cells and add 1.2 microliters of 2.5 mg per milliliter calcium-dependent Oregon green dissolved in DMSO for a final concentration of 10 micromolar. Incubate the tube for 40 minutes at 37 degrees Celsius and 6% carbon dioxide. Using a 200 microliter pipetta with a P200 tip, remove all of the solution from the tube. Completely fill the chamber by gently injecting into the inflow port. Tilt the chamber so that bubbles escape through the outflow. Place the entire chamber into the incubator and incubate for a further 20 minutes at 37 degrees Celsius and 6% carbon dioxide to allow the sperm to adhere to the poly-D-lysine. Mount the chamber to the inverted fluorescent microscope and connect it to the perfusion apparatus. Leave the preparation in the dark for at least 10 minutes to let the perfusion sweep away loose cells and extracellular dye. Under phase contrast at 40x or 60x magnification, choose a field of view where cell spacing is sufficient to permit fluorescence from each cell to be assessed separately. Cells should be adequately attached, but preferably exhibiting clear flagellar activity. Switch the microscope to the green fluorescent channel. Keep exposure times at the minimum required for a clear fluorescent image to reduce photo-bleaching. In IQ software, activate the time series image acquisition. Acquire images at 0.1 Hz and use a software-controlled shutter to limit illumination, initially recording for a control period of 3 to 5 minutes. Manipulate the saline's calcium concentration and apply drugs by changing saline at the header of the perfusion system. Note the time that each stimulus is added to the header and calculate its arrival time in the chamber as described in the written portion of this protocol. To analyze images offline, use one of the drawing tools to outline a region of interest, or ROI, around each cell or part of a cell, here the posterior sperm head because stored calcium is mobilized in this area. Scroll with the slider bar to check the image series. Identify and remove cells that fail to remain fully within the ROI, overlap other cells, or have shown the large and rapid increase followed by sharp drop-off of fluorescence intensity characteristic of cell death. Use the Analyze function to generate a time fluorescence intensity series for each ROI. The data can then be exported and analyzed in a spreadsheet. These images are from a series obtained during stimulation with 3 micromolar progesterone. The lower row has been coded in pseudocolor to better illustrate relative intensity changes. Progesterone entered the chamber here. Here, these results are animated in a time-lapse video. Notice the rapid spike in fluorescence intensity followed by a slow increase, indicating corresponding changes in intracellular calcium levels. These data are quantified here from average intensities of the posterior head of 3 cells. 3 micromolar progesterone was added at the time marked by the arrow. The dashed line shows mean control fluorescence.
Today we've just shown you how to isolate good quality cells, load them with a calcium sensitive dye and monitor the response of individual cell to stimulation.
When doing this procedure, it's important to remember to use the best possible cell population. Also, minimise illumination of the cells to avoid photo damage and pick the dye that suits the responses you're looking at best.
So that's it. Thanks for watching and good luck with your experiments.